I took my lucky break and I broke it in two.
John, a young and single but gay man, travels to other cities far away from home and his family to meet other gay men or partners. His family does not know about his homosexuality. He began to fall sick on a regular basis and avoided work for days. He visited his doctors and was later diagnosed with HIV. When his family was notified, they stopped visiting him and abandoned him because they feared that they would also become infected when they came in contact with him.
I took my lucky break and I broke it in two. Put on my worried shoes, my high worried shoes. Took me so many miles and they never wore out. My worried shoes.
Ivy, an unemployed 39-year-old woman, was pregnant with and diagnosed with HIV. She recently returned from Africa and has an 8-year-old son. She practices safe sex and tested her HIV status on a regular basis. She remembers only one day when the condom broke on her. Upon being tested, she finds out that her CD4 plus count is 1,130 and her viral load could not be detected. She discontinues taking her medication since the doctor cannot verify if the drugs will have any side effects on her unborn child. She also avoids taking AZT at this time of her delivery. At birth, Ivy's baby was tested HIV negative.
My high worried shoes. I made a mistake that I never forgot. Tied knots in the laces of my high worried shoes. Every step that I take is another mistake. I march further and further away in my high worried shoes.
Advised by her doctor that she would have to begin taking HIV medication because she's experiencing an increase in her viral load and decrease in CD4 cells. After a week on Atripla prescribed by her doctor, she suddenly experiences side effects such as depression, dizziness, and abnormal dreams. She decided to discontinue taking this medication, but upon sharing this with her support team, she was advised to continue the drugs because those experiences would only last for 2 weeks. She continued taking them, and after a visit to her doctors, she shared with her group about the increase of her CD4 cells in the 450s and her viral load was less than 95. Okay.
Go.
Hi, my name is Joey Printon, and I would like to talk to you today about the pathophysiology of HIV and AIDS. HIV and AIDS is a retrovirus, meaning it needs cells from a host in order to make more copies of itself. We call this replication. A retrovirus is something that takes its RNA and inserts it into the DNA of the host cells. These are called CD4 cells, so that therefore it can replicate and via mitosis and spread throughout the body. HIV attaches to CD4 cells, allowing the virus to enter and infect these CD4 cells, damaging them in the process. The fewer CD4 cells your body has, the weaker the immune system, and therefore the more vulnerable one is to contracting illness and infection. Here's a brief schematic of the process of HIV and AIDS. As I mentioned before, viruses lined with spike glycoproteins come and bind to the receptors of the host cell. These glycoproteins open up the cell membrane of the virus and insert its RNA and DNA into the cytoplasm of the cell. The viral RNA mixes with cell RNA to form DNA, which is then sent into the nucleus of the host cell. The host cell, or once the RNA from the virus is implemented into the host cell DNA, it causes the cell to produce more of this bad RNA that came in from the virus. The virus then secretes this RNA out of the nucleus, and once again, it is reestablished and is excreted from the cell, moving on to infect other host cells in the body.
My shoes took me down a crooked path, away from all welcome mouths. My worried shoes.
While there is no cure for HIV or AIDS, the main type of treatment or therapy is known as the antiretroviral treatment. Now, this treatment consists of drugs that must be taken every single day for the rest of one's lives. The goal of antiretroviral treatment is to lower the amount of HIV in one's body. This would stop the weakening of the immune system and perhaps reverse any damage that has already been done by the disease. As far as classification of these drugs go, there are 5 main types of antiretroviral drugs that are used. 1 is nucleoside reverse transcriptase inhibitors. 2, non-nucleoside reverse transcriptase inhibitors. 3, protease inhibitors. 4, fusion or entry inhibitors. And 5, integrase inhibitors.
Go.
The use of 3 or more of these drugs at the same time is known as HAART, or highly active antiretroviral therapy. Although it is considered the best therapy of our time, doctors and scientists are undergoing a new therapy using zinc finger nucleases, where they would go into a specific domain, pull out a specific genetic sequence, rewire it, and fix the HIV, and simply put it back in one's genes. Although human testing has not begun, animal and plant testing has, and hopefully it will be a cure for the future.
Ooh.